So you're doing a split screen with our textbook, Chapter 10, Section 5, and then over at Notability, Ribosomes and Protein Synthesis. Let's go down to the second page, the top of the second page. It says go to Chapter 10, Section 15. That's where we're at. We're going to summarize what transcription and what translation is in this box. So here's transcription in purple. What's the main idea of transcription? You sketched it out in your homework in the last class. It's right there in the purple box. What's the goal of transcription? Really? Everyone's drawing a blank? Or are you just shy? Great. The word script or transcribe means to copy. So what are we copying from and what are we copying to? You played the protein synthesis race. What's the first thing that you did in part 1? It was called transcription. Yeah?
You match the letters.
Right. And so we are taking the letters from DNA and putting it into RNA. It's really a copy, right? And the reason why is because DNA can't leave the nucleus. And the information in DNA is needed in the cytoplasm where the ribosomes are. And so we are going to leave DNA where it is in the nucleus, but take a piece of the code in DNA and write it into RNA. Yeah?
What were the two words that means copy?
Transcribe.
And translation.
Yeah. No. That's the second step. If you're talking about making a copy of DNA into RNA, I think I called it the actual end product as a noun. It's called the transcript, and the process of copying it is called transcription. They're all the same word, right? And that base word is scribe, which means to copy. So it says, how are you going to keep track of this? Complete the table with the main idea, and that is we need a copy of DNA, but DNA can't leave the nucleus. So we're going to make a copy of it in a single strand, and that's what RNA is. So if you're fuzzy with this, you're just confident that the test isn't this week, so you don't have to worry about it. But when your classes get tough, and for some of you this class is tough because struggling to get a C, you have to realize your homework includes understanding what you're writing down. When you get to a class and when you get to upper level class in college, you can't let 2 days go by or you'll never get caught up. You have to stay on top of it. So I'm here to show you that your one page that you can use in your textbook is right here. This is a summary page. Look at the top, it says review. The flow of information from DNA to a protein, it's a central theme in biology. You go from DNA to RNA, that's transcription. And then when you go from RNA to protein, that's translation. So over here in your homework, transcription is going from DNA to RNA. You're copying the 4-base code. In translation, you're going to do what Ralphie just did in A Christmas Story. You need to have a translator wheel. You're in Spanish class, you're in German class, or maybe sign language. When you learned to go to another language, you needed a translator. You had to have a dictionary that said, all right, this is the English word, this is the Spanish word, this is the German word, this is the English word. So there's a translation that's going to happen, and we're going to go, now we're over here at translation. The main idea is we have to go from the nitrogenous base code, we have to go from the 4-base letter code to the building blocks of proteins. What are the building blocks of proteins? What's the end result? So I'm at the bottom of the picture, and it's labeling it with this purple chain right here. What is a polypeptide? You might not have heard that term before, but when you did the protein synthesis race, what were you making? Did you make sugar? Did it make fats? Did it make proteins? Yeah, that's why it was called protein synthesis race. Speed didn't matter, right? But you had to get to a protein. So over here in translation, you're translating from the 4-base code to proteins. What are the building blocks of proteins? What is this square? What is that triangle? What is that circle representing? What are the building blocks of proteins? You sketched them as little circles on your paper. Yes?
Amino acids.
Correct. And so up at the top of the page, it even says, hey, these are amino acids. So didn't that little Ralphie video clip help you to realize that he had a code that he got off the radio? That's like your nitrogenous bases. That's like the code in messenger RNA. He was writing it down. Then he needed that code ring, and they said, set your decoder to B2, because if you spin that wheel, every letter and number would be something different. So we're translating from nitrogenous bases to sequence of amino acids. And that's a big takeaway right there. So making proteins is a 2-step process, right? Transcription, which happens in the nucleus, because DNA can't leave the nucleus, it's too big. And then here's the cytoplasm. That line would be the membrane that you drew in your sketching picture. And the messenger RNA goes through that little pore, goes down into the ribosome. You sketched a 2-part ribosome, right? A large top piece, like a top of a hamburger bun, the bottom of the bun, and then the messenger RNA becomes the meat and the cheese, right? It's the important part. Then you also learned that every 3 letters is a code, and we call it a codon. So messenger RNA, every 3 letters, every 3 bases are read together. Well, we know that the purple piece up here at the top of this transfer RNA, they don't label it here in this picture, it's up a little bit higher. Every transfer RNA is bringing in an amino acid. So there's a place inside here to hold 3 transfer RNAs all at one time. So if you look at the picture, it's showing 2 of them right here. So the ribosome they called it the factory. This is where proteins are going to be made. But in this picture, there's 3 types of RNA, isn't there? And in your notes the other day, you sketched it. Messenger RNA, transfer RNA, and the ribosome itself is RNA. So this is mRNA, this is tRNA, and this is rRNA. Now, if you don't practice this, then you're always wondering, hey, can I do this on the quiz? Oh, I'm really thankful it's multiple choice because I can guess my way through it. No, you can't. Statistics are way against you. You have to practice it like you're having to be an essay test. And that's what office hour is really good for. Sit down with a partner and run through it. And if you do this days or even a week or two ahead of your quiz, you'll get an A in your upcoming quiz because you're just prepared. If you wait till the last minute, it rarely comes together. So, how does the transfer RNA know how to come in? Right? They're base paired. The codon on the messenger RNA matches up with the anticodon on the transfer RNA. They complementary base pair. C goes with G and A goes with U. Remember that exception? There is no letter T in RNA. It's replaced by a U. So I just said something that's like not on the worksheet and it may have gone over your head, so let's write that down. Do you know you're responsible for writing yourself notes for when you don't understand something? So let's go back up here now. I'm going to write down in DNA, there's a letter C, there's a letter G, there's a letter A, and there's a letter T. In RNA, there's a base of C, there's a base of G, and there is no base of T. It's replaced by a U. So these are the base pairings. There's no U in RNA. A week ago, a student said, hey, what are we covering next? I want to take a look at the textbook chapter. So they're even ahead of me. And when you do that, you're an A student. You're going to get A's because you're spending 5 minutes, right? 10 minutes. So there's a big pendulum swing. Yeah, that person is really got their study skills down. And then you swing the pendulum all the other way around, and some people never open up the textbook. So yeah, you can find a happy medium, and usually it's 5 minutes, 10 minutes. And the best way to do that is during class. So if we're talking about protein synthesis, why not open the textbook up and be checking that out instead of watching videos and playing games on your iPad. So there's a direct correlation to how much you goof around on your iPad during the school day and the type of grades that you get. And the faster you figure that out, the better your grades will be. And if you're an athlete, in our WASP meeting this morning, I heard that 75% of students are athletes. And if you want to go play at the collegiate level, colleges really want you if you are a B or an A student. If you're a C or a D student and you want to play in college, you might have a chance to walk on and try out, but you're not going to get recognized because colleges are afraid of C and D students not making it through one semester, so they don't want to invest in a playing spot for someone that doesn't have their academics down. So you have an awesome opportunity to be a scholar athlete here at Modern Day. There will be hundreds of students in your freshman class that will graduate with a higher than a 4.0 GPA. So if you've struggled this year, get back on track because you are having such an opportunity to be a great athlete because we have super coaches. And then just get your study skills down. Come on in to afternoon office hours and there's very little distraction. And you can get a lot done in that time. And so that's really still the school day. All right. Go ahead and fill in this up here. Write down, what is an anticodon to you? What is a codon to you? What is gene expression? How are you going to remember the word polypeptide? Take a couple of minutes to fill in how you're going to memorize that. Do some sort of memory trick. Remember I was telling you about the car? If you had to memorize a list of things, you're going to put something on the steering wheel. You're going to put something in your backpack, something's in your wallet. Or do a play on the name. The codon. This is Ralphie's code wheel. That's on messenger RNA. The anticodon is on transfer RNA. It matches up. It's the Antichrist. It's the opposite. The more outrageous you can make your analogies, the easier you're going to remember it. Anti means opposite. It's the opposite of the code on the codon wheel. I thought of this one at lunch. You know what poly stands for? Yes? Many. And that word looked like Pepsi to me. So I thought, what if you had 20 Pepsi cans all in a row right here? That would be a poly Pepsi. So what does that mean? All the building blocks are amino acids. When you bind them together, they become a dipeptide. 2 Pepsis. 3 amino acids, 3 Pepsi cans. That's a tripeptide. Then when you have more than 3, it's called poly. So you got to think of creative ways to remember things with outrageous images and things like that. So when you're studying, sometimes just how am I going to remember that is as important as just trying to look at it and go, how am I going to remember that? Like, what's the order? You come up with some way of remembering it. So we went over all of these when we were looking at page 15 just a second ago. So go ahead and fill those in. Fill it in the way you're going to remember it. If I tell you what to write down, it works for me, but maybe not for you, so you got to come up with your own way. And proteins are usually thousands long, so imagine 1,000 Pepsi cans just rolling around the room, and they're all touching each other because they've all had the opportunity to be in the ribosome together, and they got bonded together, twisted together. So you owe it to yourself to look over chapter 10, page 15. Believe it or not, we have covered pages 1 through 14 already, just in our video and you sketching. And in science, a lot of what we talk about is a process. It's a sequence of events. And so wasn't your stop motion video really good for repetition for you to get to understand something? So be on the lookout when I grade this. If you missed something, that made it confusing, I might say redo this within 1 day to get a higher score, because I want you to have a good win here to raise up your grade on this take-home quiz. So now let's practice this, because we've seen it 3 times now already. You sketched it. You did the protein synthesis race. We just looked at it on page 15. And now a 4th time we're going to look at it here. And the magic number is usually 7. You got to hit repetition about 7 times in order to start making it make sense. So if I go over it 4 times in class and then you stop and don't do it 2 or 3 times yourself, then you're going to have chinks in your armor. Something's going to confuse you. So it does take some practice. Circle each codon. How many bases is a codon?
3.
So you're going to circle 1, 2, 3. See if you can answer questions 2, 3, and 4. If you don't know a word, you have to break it apart and you have to spend some time defining it.
My pencil isn't working.
Use your fingers then.
Can I go to the tech?
Oh, is it a dead?
It's like
Is it dead?
it's charging, it's connected, but just the tip isn't working. And it's like something I said.
If you can go really quick, but I wouldn't stand in line and miss class. So I'm going to define, what does poly mean? Poly means many. And I'll define what a peptide is for you. A peptide is the bond that holds amino acids together. It's such an important bond, it has its own name. Yeah, it's a covalent bond, but the word peptide means that it's holding 2 amino acids together. So when you say that something is a polypeptide, you are talking about a bunch of amino acids bonded together, so you are describing a protein, which is a chain of amino acids. Did you get the answer right? What does the letter A stand for in the genetic code? Adenine. Adenine? Yep. Can a codon contain 2 of the same nucleotide bases? Like, could it go AAC?
Yes.
Yeah. You saw that in the protein synthesis race. This is called a codon wheel. You can see that it's a wheel. What did they say?
Oh, this is actually the acid. I want to replace it with afterwards.
Oh, really? Cool. Write the name of the amino acid that corresponds to each messenger RNA code. So here's Ralphie's decoder ring. That was a really good video. Have you seen the whole movie? Pretty cool. So how do you read AAA on this wheel? You read it from the inside out. So codons are 3 bases in a row. So A is right there, A is right there, and A is right there. So it says that it's lysine. So the first one was done for you. So I'll put a letter. This is the first base. This is the second base, and then the third base is the outer wheel. So GCG, you look at the inner G, and then the C, and then a G, and so it's alanine. So this column right here is the amino acid that the transfer RNA is going to have with this messenger RNA. So this is how you decode DNA and RNA. What we're doing right now, is this transcription or is this translation? What is it? I didn't hear it. Translation, right. We're translating the code to an amino acid, and that's translation, and this is the translator wheel. So how'd you do on the other ones? GAU. GAU, it is aspartic acid. And the last one, CAA, glutamine. It works out pretty nicely that they abbreviate all amino acids by their first three letters. So this would have been abbreviated ALA and ASP and GLU and LYS. So now we're looking for histidine. There's histidine. So histidine on the wheel is right here, and it would be CAC and CAU. CAC and CAU. Somebody called out 4 for question 3. How many different messenger RNA codes correspond to arginine? So I'll highlight that one different color. Arginine is right there and over here. So the answer isn't 4, is it? How many different messenger RNA codons correspond to the amino acid arginine? There's 4 here. CGG, CGA, CGC, CGU, and then there's AGA and AGG. So the answer is pretty obvious, 6. Here's methionine.
1.
There's only one code for methionine. And you know, interesting, this codon starts every gene. Every gene will start with methionine, and every gene will end with a stop codon. So look at the wheel. There's a stop right there. And there's 2 stop codons. No, thought there was, yeah, there's 3 stop codons. UGA, UAA, and UAG. So what does the stop codon mean? When it gets to the stop codon right here, it's going to let go of all of these and let all the proteins go. And it's not going to add any more to it. Oh yeah, they labeled the stop codon right there. So this isn't too hard, right? Especially if you were paying attention during the protein synthesis race. I'll get you started on the translation. So this is messenger RNA. You can make it a little easier by writing up above here that this, the anticodon, this is the codon, and now I'm going to write the anticodon. It would be U, UAC, AAG, UUU, GAC. So now you can see that leucine is going to go right there. Lysine is going to go right here. Next week, I've got all of these like on pieces of paper, and I'll have magnets up on the wall, and you guys are going to take turns coming up here and transcribing and translating. So it just takes a little practice. I got a little messy the way I wrote it. Which anticodon matches the messenger RNA codon UUC? So remember the base pairs, C goes with G and A goes with U. It's not too hard to remember those, just two things to memorize. So if you're a musician, you know that the chords C and G go really well together. They're a fourth or a fifth, depending on which direction you're going. And so musically, it sounds good. So to me, those are the ones that I remember. And then the other one, A and U. Remember the Fonzie on Happy Days? Hey, how are you? So what are some ways to remember this, you know? You have to come up with ways of remembering sets of things so that you don't miss it. If you're having trouble memorizing something, just sit down and go, how can I remember that? Let me do imagery. Let me set this on the dashboard of the car. Let me come up with an acronym. Something from a movie? If you don't think you have a good memory, just Google it. You'll find a lot of ways that you can help to memorize and keep track of stuff. And today's students are a lot different than when I was in high school. We had to do a lot of memorizing because we didn't have computers, basically. Had basic computers. They just did DOS and simple basic programs. There was no Google. You had to go to the card catalog and look up books in the library. Then you had your information. Then you had to try to memorize it. You're in the day of age when you just have to be able to Google something. So what's really frustrating is when somebody emails me and said, "Oh, I didn't know how to do that." And I'm like, seriously? You're holding a computer that powered a spaceship to the moon. I mean, you've got all this computing power, and you just say, you sent me an email saying you didn't know how to do it? That's where your power is, knowing how to search information really quickly because it's right there at your fingertips. Which amino acid is carried by the anticodon UUU? Lysine. So be on the lookout for this. It says which amino acid is carried by the anticodon. That means that it's on the transfer RNA. And so here's UUU, and it's carrying lysine. You just have to be careful. Are they asking for the codon or are they asking for the anticodon info? So number 4, list the amino acids in the order they would appear in the polypeptide. What's a polypeptide again?
The chain of amino acids.
Right. All these Pepsi cans listed in order. So it would be METH. PHE. LYS. LEU. So I'm taking the first 3 letters of the transfer RNA. So the first one, MET, methionine, always starts the genes. I could have just done MET. And then number 2, PHE, phenyl. I was thinking it was phenolphthalein, but phenylalanine. There are only 20 amino acids, and you know, some of you guys who are athletes, you work out and then you're going to grab some protein powder, because if you eat protein powder, it can help build up your muscles. And you look at the label, it'll say filled with 20 amino acids. I even have seen a liquid source of amino acids. So you can't build your muscles without having the building blocks of muscles, building blocks of proteins. That's what your muscles are made out of, or amino acids. So it would be great if you were to look at this again before our next class, and what you would do is you would start with a blank one and see if you could fill the whole thing out. Don't wait until next week. You should do it this week, and if you do, it's going to all make sense to you, and then you're filled with confidence. Being a great athlete is all about confidence, right? Tom Brady said he was going to retire, and now he's saying he's going to come back because he's confident that he can win another Super Bowl. Every athlete at the top of their game, they've done a preparation, and they know that they know it, and they know that they're the best. So it just takes practice. You'll get there. Any questions with this? I meant to check during office hour if this is available to you. It wasn't last year, but I think you guys have upgraded iPads. So can somebody check this out for me? It's called Protein Synthesis Virtual Lab. can you click into this assignment? This is homework, and I think it's going to run on your iPad. It runs on mine, and I think you guys got upgraded ones. So go to this website, and you're going to go down here to transcribe and translate a gene. Go down and click into transcribe and translate a gene, and I think it works for you guys. If not, it's going to be homework for you to do it on a computer at home or a computer at our library, but let's see if it works for you. So once again, go over to the assignment Protein Synthesis Lab and click on that link. And then you have to click on transcribe and translate a gene and see if it works for you. Yeah. Okay, good. And it's going to tell you what to do on the screen.
Wait, where? It's the virtual lab.
It sounds like the word code, right? So it's the secret code. Every 3 letters on the manual.
Where's the video at?
Oh, look at the washing.
Is it the link?
Yeah.
All right, it's working on your iPad. So for the remainder of the class, go ahead and that is your homework. By the way, what we just finished is also graded, so you have to turn that in. I think you saw that at Canvas.